How pharma companies prove sameness to the FDA. The Liraglutide playbook. A regulatory team submitted an ANDA for the peptide-based drug. The analytical package was thorough by most standards. 6 months later, FDA issued a complete response letter. The data existed, the interpretation of sameness was not there. That's the difference this video is about. Hello, I am Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories. We specialize in the analytical and regulatory characterization work that sits between a well-made generic and an approved one. Let's start with why liraglutide, the active ingredient in Victoza and Saxenda, is not a molecule you can prove identical with standard small molecule methods. Liraglutide is a 26 amino acid GLP-1 analog that alone puts it in a different analytical category. But what makes it genuinely complex is its acylated side chain. A C16 fatty acid attached at lysine 26 through a gamma glutamic acid spacer. That modification is what gives liraglutide its long life, and it's also what makes proving sameness a layered structure problem. A less experienced team might confirm the peptide backbone and consider the job done. The lipid attachment is where the real work begins. So what does FDA actually require under an ANDA for a complex peptide like this? The standard is molecular level sameness, not just equivalent clinical effect, but demonstrated structural identity with a reference listed drug. This means the generic applicant must show that the active ingredient is the same as the reference in terms of molecular structure, impurity profile, and degradation behavior. FDA guidance on complex drug substances is explicit, where the active ingredient contains structural elements that contribute to biological activity. Each must be independently confirmed. For liraglutide, the acylated side chain is not a footnote, it's a critical quality attribute. This is where many development programs underestimate the scope. Why is sameness testing for liraglutide genuinely difficult? 3 reasons. First, molecule structural complexity requires orthogonal methods. No single technique confirms everything. Second, impurity profiles for acylated peptides can include subtle variants, incorrect acylation position, alternative lipid chain lengths that are analytically close to the correct structure but pharmacologically distinct. Third, the degradation behavior of liraglutide under stress is multifactorial. Oxidation at methionine, deamidation, and hydrolytic cleavage can each produce degradants that would fail a sameness argument if the generic and reference pattern diverged. Early in a project, a client sent us a liraglutide drug substance lot alongside the reference product for parallel characterization. At the same intact mass level, the two looked indistinguishable. But when we ran peptide mapping with complete sequence coverage, we identified a minor acylation variant in the generic lot. The fatty acid was attached at the different lysine residue. The difference was sub-percent. FDA would have found it. We found it first, which meant the client had time to investigate the root cause in manufacturing rather than receive it as a deficiency in their submission. The analytical strategy for proving sameness follows a deliberate sequence. LC-HRMS confirmed intact molecular mass. This is your first gate, and it needs to be clean. LC-MS then verifies the complete peptide sequence at the fragment ion level. Peptide mapping achieves full sequence coverage, which matters because partial coverage leaves the regulatory questions open. Then orthogonal methods, often including circular dichromism and NMR for higher order structure context, reduce uncertainty at the points where a single technique would be ambiguous. Stress testing under force degradation conditions rounds out the picture by mapping how each molecule degrades under the same stressors and whether the profiles align or not. Verifying the lipid side chain specifically requires confirming four things: that the fatty acid is palmitoyl C16, that the attachment at the lysine 26 and not at the adjacent residue, that the gamma glutamic acid spacer is intact and positioned correctly, and that incorrect acylation variants are excluded, not just absent above a threshold but systematically tested for. This last point is where data interpretation matters as much as data generation. As a result of not detected is only meaningful if the method was sensitive enough and specific enough to detect the variant if it were there. Impurity and stability comparisons close the argument. Oxidation and deamidation trends between generic and reference must track. Thermal and photostability study results must align. Hydrolytic degradation behavior under pH stress must produce the same degradants at the comparable levels. When there are no unique degradants in the generic and impurity profiles mirror the reference, you have the foundation of a defensible sameness cases. None of this analytical work carries weight without documentation built to survive regulatory review. CTD compliant format is the minimum. LCOA plus data integrity principles, attributable, legible, contemporaneous, original and accurate is the full forms of LCO plus, must be woven into how data is generated and recorded, not retrofitted. 21 CFR Part 11 compliance for the electronic records is non-negotiable. When an FDA reviewer opens your Module 3 analytical section, the data package should answer questions before they asked. Here is what to take from this. Sameness for liraglutide is not a checkbox, it's a structural argument built method by method. Impurity alignment is as important as molecular identity. Force degradation is not optional risk mitigation, it's a regulatory expectations, and quality of your documentation determines whether a strong analytical package translates into smooth review or a CRL. If you are working on a liraglutide ANDA or evaluating the analytical scope for a complex peptide generic, we have worked through these questions before. Reach out to Resolve Mass Laboratories and we would be happy to have a conversation with. Thank you so much for watching this session and see you in the next one. Until then, goodbye for now.